Please listen carefully.
Hello and welcome to GasGasGas, the podcast that covers the FRCA Primary exam. We're going to fit into your day and give you as much of your life back as you could possibly imagine. I'm here to make your studying easier. Listen to us on your commute, in the gym, in the shower, or when you're ironing your scrubs. Expect facts, concepts, model answers and the odd tangent. Check out the show notes for all the detail and remember to follow the show so that you never miss an episode. Let's get on with it. Hello Tom and welcome to your physiology viva. Obviously we're smashing through these at a tremendous rate. But most importantly, what do you reckon about this cardigan I got on, eh? Although it's a bit hot. One sec.
Hey, why would you buy a cardigan and then pull it over your head?
Hey?
You're an animal. Yeah, that's the whole joy of a cardigan. And why not go through the intermediate process of unbuttoning and see if that's a nice balance? You don't deserve a cardigan.
Oh, I didn't realise you felt so strongly about cardigan donning doffing techniques, Tom. Sorry, I'll buck up my ideas. Anyway, focusing back on the task at hand there, Tom. How do you think your physiology viva question answering techniques have been going?
I don't even know how to answer that question. It's, I think I vaguely feel as though I know lots of physiology and then I keep getting asked questions where I go, Not.
Yeah, how do you feel about the Nernst equation?
Oh, I do not know the Nernst equation, and I can only vaguely recollect what it's useful for. I did listen to
Fortunately enough.
I was just going to say, I listened to some neuroscience lectures before medical school from MIT by a really monotone neuroscientist over there, and he, I think, was involved in all of the squid neuron experiments they like to reference, and he was very much a fan of the Nernst equation and used to love talking about Acceleronstoff, which I think is the original German name for adrenaline.
That's some niche knowledge, Tom. Unfortunately, I've not done Nernst equation questions for you today, but maybe next time.
Nice hint. I wish they'd do that for the exam. I suppose they have actually. They wrote a syllabus, didn't they?
They did. It's just rather broad.
Yeah.
Righto. So, are you ready for your timed 3 5-minute stations physiology viva?
Yeah, I'm always ready.
Righto. So, Tom, welcome. This is your physiology viva. What are the physiological effects of the sudden loss of 1 litre of circulating volume?
When a large volume of your circulatory volume is lost, it has some immediate effects, intermediate effects, and delayed effects. The immediate effects are for baroreceptors within the carotid body and the aortic arch to sense that pressure within the vascular system has dropped. This leads to activation of the sympathetic nervous system via central pathways. It leads to secretion of antidiuretic hormone from the posterior pituitary, which promotes reabsorption of fluid from the distal convoluted tubule. And so backtracking a little bit to more immediate effects, you have an increase in heart rate via that sympathetic activation, which helps maintain blood pressure. You have increase in stroke volume. You have venoconstriction, which moves blood from capacitance vessels, such as within the liver, to try and replace the circulating volume that's lost. And you have peripheral vasoconstriction from that sympathetic activation as well, by action at adrenaline receptors in smooth muscle. All of these things help to maintain blood pressure. The intermediate effects involve retaining fluid. So I already mentioned secretion of antidiuretic hormone, just because I was thinking of the neuronal pathways that link to the central nervous system. But on top of this, there will be drop in blood pressure detected by the perfusion to the kidney. So by having reduced venous pressure in the within the venules of the kidney, you get reduced sodium flux at the macula densa, which leads to release of renin from the juxtaglomerular apparatus, the secretion of aldosterone, and that promotes fluid retention in order to try and maintain blood pressure. This the same system, so the secretion of renin from the juxtaglomerular apparatus, as well as promoting the release of aldosterone, will lead to the activation of angiotensin, to conversion of angiotensin 1 to angiotensin 2, in fact, which is a potent vasoconstrictor and will itself lead to further activation of the sympathetic nervous system and secretion of catecholamines. They're the mechanisms that spring to mind immediately.
And any other fluid shifts? You mentioned capacitance vessels, but is there any other store of fluid that can be mobilised?
Yes. So, yeah, I suppose we talked there about immediate and intermediate actions rather than the delayed ones. So, fluid will move from the interstitial space into the vascular space to replace that fluid which is lost, and it will do that in response to reduced hydrostatic pressure in the vessels of within blood vessels essentially, and the reduction in Starling's forces. So as that fluid shifts across, it will also draw fluid from the intracellular compartment as well to some extent, and lead to replacement of the lost volume to a point.
Have you got any formulaic way of framing that shift of fluid?
So I can talk about the percentage blood loss compared to the physiological effects, and I can give an idea of what percentage of total body fluid lies within each compartment, but I haven't.
I was trying to get towards the Starling equation.
Oh yes, I mentioned Starling's forces there, so it would be useful to draw a diagram here, but
what factors are there?
Yeah, so the diagram I'm thinking of really just summarises the fact that there is hydrostatic pressure within blood vessels and there is osmotic pressure within blood vessels, and the same exists in the interstitial space around them, and the balance of those forces decides whether or not at a capillary level fluid shifts into or out of that space. When we lose volume from the vascular space, we get a reduction in hydrostatic pressure, as I mentioned earlier, causing fluid shift inside, but also as fluid shifts into the vascular space.
And that's your 5 minutes, Tom. We're going to move on. How would you define the resting membrane potential? What is it?
The resting membrane potential is the potential difference across a cell membrane in normal physiological conditions when electrolytes are sitting in equilibrium either side of a cell membrane. So a neuron resting membrane potential is normally around minus 40 millivolts, and it's dictated by the concentration of cations and anions either side of the cell membrane. Through the action of potassium ATPase pumps, primary driver behind resting membrane potential will be the raised concentration of potassium within the cell and the raised sodium concentration outside of a cell.
Could you just elaborate a little bit further on how those ion differences are maintained?
Yes, so the sodium potassium ATPase pump is a transmembrane protein that utilises adenosine triphosphate in order to consume energy and to move sodium and potassium in opposite directions against their concentration gradients. So potassium moves within the cell and sodium moves out of the cell. This sets up an electrochemical gradient and results in a resting membrane potential.
Okay, and what triggers depolarization of this cell, this nerve cell, for example?
So depolarization of nerve cells is often thought of as an all or nothing response. It begins by reactions typically on the postsynaptic membrane of a neuron. So a preceding neuronal cell, under the influence of various factors, can release neurotransmitters into the synaptic cleft between neurons, and when they do this, they cause downstream effects at the postsynaptic membrane in the subsequent cell within the chain. Typically, these reactions lead to the rapid influx of sodium in response to binding of neurotransmitter to a receptor on the postsynaptic membrane and efflux of potassium. There's also important function of calcium release intracellularly, normally stored mainly in cytoplasmic reticulum, but this mechanism varies a little bit from cell type to cell type.
How is it conducted down the nerve?
So action potentials are conducted down the nerve via decremental conduction and do so with saltatory conduction, so they have rapid propagation of voltage-dependent opening of sodium channels and a rapid change in the resting membrane potential, which jumps between so-called nodes of Ranvier in myelinated cells, and in demyelinated cells there is slightly slower decremental conduction down the length of the neuron, but still via voltage-dependent sodium channels.
And can a nerve that's been depolarized immediately depolarize again?
No, so after depolarization of all cells that depolarize, there is an absolute refractory period and a relative refractory period. During the refractory period, the absolute refractory period, it's impossible for a cell to depolarize again, no matter how strong the stimulus from the postsynaptic membrane that normally starts decremental conduction and depolarization. In the relative refractory period, you can produce further conduction, but you need a supranormal stimulus in order to do so.
Yeah, you mentioned there that some nerve fibers are myelinated and some are not. Could you just classify those peripheral nerve fibers for me?
So we could name some of them via the parts of the nervous system they affect. So autonomic nerves are unmyelinated, which means they have slower conduction but are particularly susceptible to uptake of local anaesthetics, for instance, hence early effects on the sympathetic nervous system during spinal and epidural anaesthesia. We have motor nerve fibres, which are myelinated in order to facilitate rapid decremental conduction and coordinated contraction of muscles. The same applies to primary and secondary neurons within spinal reflex pathways. They are also myelinated. We then have sensory fibres travelling through the spinal thalamic tracts to the brain. So there are different types of nerve fibre, such as C fibres and delta fibres. So C fibres typically are unmyelinated. That's as much as I can remember now.
Oh, okay. And actually, as we have it, that's your 5 minutes on that topic area. Oh, yeah, I think resting membrane potential of a nerve there, Tom, is minus 70 millivolts.
But yeah, that was another good example of physiology that seems familiar, but it's woolly. I've not gone over it in a while. Minus 40 is, I'm thinking of the automaticity, unstable membrane potential in cardiac pacemaker cells, aren't I?
Yeah, minus 70 for a nerve. And I think you said osmotic pressure for Starling's forces. It is oncotic pressure.
Oncotic, absolutely. Yeah, slip of the tongue, that.
Right. Okay. And this is your last 5 minutes because I'm now organised here, Tom. And it's one of your favourite topics, as I understand it. Can you define pH?
pH is the negative log to the base 10 of the hydrogen ion concentration in an aqueous solution.
Okay. And what is this relationship between hydrogen ions and pH?
So the relationship is defined by the equation I've just described, but its pH scale itself really is just a way of expressing hydrogen ion concentration. The relationship between specific chemicals and the resultant hydrogen ion concentration can be described by the acid dissociation constant. So acid dissociation constant is the division of the conjugate acid over the conjugate acid concentration times the conjugate base concentration, and the negative logarithm of this is known as the pKa.
Okay. Well, talk me through the Henderson-Hasselbalch equation.
So the Henderson-Hasselbalch equation relates the acid dissociation constant with the concentration of conjugate acid and conjugate base. So the pKa is equal to negative log to the base 10 minus the concentration of conjugate acid over the concentration of conjugate acid times conjugate base.
How is normal acid-base balance maintained in a human?
So acid-base balance can be divided into rapid, intermediate and delayed homeostatic responses. The rapid responses I would group buffer action and respiratory action together there. So when hydrogen ion concentration increases in the plasma, buffer systems immediately act to stabilise the pH of a solution. So a buffer just is a weak acid that moves towards, well, it's in equilibrium between its conjugate acid and conjugate base, and by Chatelier's principle, it acts to oppose any change made within the system that it's sitting. Good examples would be histidine residues and hemoglobin, which absorb hydrogen ions when they're produced during CO2 transport, but also...
You mentioned respiratory compensation. What's that?
Respiratory compensation really involves the body's response to PCO2. So as PCO2 increases, pH decreases, and in order to achieve homeostasis, respiratory centres in the brain respond to increased PCO2 by increasing respiratory rate and tidal volume, thus reducing the PCO2 and increasing the pH. The delayed responses that I was talking about involve primarily renal handling of acid and bases. So in the proximal convoluted tubule, in response to reduction in pH, bicarbonate is reabsorbed, and in the distal collecting ducts, H plus ions are secreted in response to a decrease in pH. All of this together aims to keep tight control on physiological pH, which is important for the action of lots of enzymes, which are biologically essential.
So, what is the anion gap?
Anion gap is defined as sodium concentration in plasma plus the potassium concentration in plasma minus the chloride concentration plus the bicarbonate concentration. It's a theoretical construct designed to help us understand acidosis and its causes. The theory itself doesn't give an explanation of mechanism, but it does help us divide up etiologies. So mud piles is the typical.
And that is your 5 minutes there, Tom. But you've got 30 seconds to tell me what mud piles is, just for the listeners.
So mud piles tells you causes of raised anion gap acidosis. So that includes methanol, ethanol, propylene glycol, paracetamol, isoniazid, DKA. Yeah, I didn't do that in order because I'm in a hurry, but multiple causes of raised anion gap acidosis.
Yeah, causes of a raised anion gap metabolic acidosis. Because a non-raised anion gap metabolic acidosis demonstrates a source of excessive endogenous or exogenous hydrogen ions, i.e. a new source of acid added to the body. So we're just all on the same page here, listeners. The acronym we really should be learning is CAT mud piles. And the full differential for a high anion gap metabolic acidosis is carbon monoxide plus cyanide plus congenital heart failure. A. Aminoglycosides, gentamicin. T. Theophylline or toluene if you've been sniffing plenty of glue. M for mud piles, methanol. U. Uremia. D is your DKA or your ketoacidoses because starvation and boozing also causes ketoacidosis. P. Piles, paracetamol, paraldehyde. I. Iron overdose really bad, isoniazid and inborn errors of metabolism. L. Lactic acidosis. E for ethanol and ethylene glycol, and S for salicylates, i.e. aspirin overdose. If you can vomit all that out at the examiner, they'll probably be blown to Timbuktu.
Yeah, so non-ion gap acidosis means that there's a problem in bicarbonate, in inappropriate bicarbonate secretion, essentially. So renal tubular acidosis, or a lack of hydrogen ion secretion. So renal tubular acidosis, more than one type, so it can be inappropriate secretion of bicarbonate or inappropriate secretion of hydrogen ions, but also
Inappropriate retention of hydrogen ion guys or inappropriate loss of bicarb would cause the acidosis. What about if I just drank an inordinately large amount of vinegar?
Oh god, you'd have to drink a lot. Well, it's a weak acid, isn't it? So wouldn't it just help with your buffering?
I don't know. I once drank a pint for a bet. It wasn't very nice. I got terrible heartburn.
It definitely can cause profound acidosis. I know that in Bristol, someone was admitted after drinking pints and pints of cider vinegar as a home remedy, and they'd given themselves like a pH of about 7.0.
I bet they felt quite ropey.
Yeah.
Anyway, that is your physiology viva there, Tom. Nice and brisk. I quite like the, what's it you say? You say immediate something delayed effects. What's the middle one?
Immediate, intermediate, and delayed effects. It was a tip given by a consultant at work, and
that is good.
I like it as an approach sometimes, but sometimes whilst I'm saying it, I think, oh God, I'm not sure if I can actually carve it up that way. But anyway.
Yeah, it makes it sound slick. That's half the battle. Yeah, cool. Any thoughts, any questions?
No, I think I just being a bit wordy, need to make it a bit snappier, but getting there.
Yeah. Still a bit like you just got to go for the jugular and like if you're gonna say the buzzword, get it in.
Just say it. Just say it.
Just get it in there. I think with the buffering, I'd be tempted to because you kind of like fudged in. It felt like you were saying that there was extracellular, intercellular buffers, and breathing were like both quite rapid. When I'd sort of framed that those intra and extracellular buffers, the phosphates and the proteins and the hemoglobin, as you say, are like
Instant. Yeah, no.
almost instantaneous. And then you've got the respiratory effects. Then you've got your renal effects.
Yeah. I should have put respiratory in the middle. I think I planned on doing that, and then I don't know why I changed my mind.
Yeah, I would have thought so.
Yeah, I didn't really get to mention.
You could just say in order of the time it takes for them to activate, your extracellular buffers and your intercellular buffers work very quickly, and then your respiratory system tries to compensate, and then your kidneys eventually catch up, but this can take days as a timeline. Yeah. Okay, well, thank you very much, Tom. That was your physiology viva. I didn't hear a single drop of sweat land on your glasses.
Yeah, well, I did a tour in Afghanistan, and ever since I've been entirely incapable of sweating.
Okay, I think we will leave this apolitical podcast there. Well, thank you everyone for listening to what is the 16th Vivacast episode that we put together. Everyone will be happy to know that Tom did in fact pass his Viva, which gives you a reasonable reflection of what they're expecting and the standards they're after. It's always important to have some structure. I think the immediate, delayed and late effects you can probably shoehorn into most things. Have a few things you can fall back on as structures, like pre, intra, post-operative. Have a few buzz phrases to open up questions, to give you a chance to think. And sometimes, very briefly, repeating the question back to the examiner gives you a summarization in your brain, which lets you start talking. You can then open up with a phrase and then say, and I like to structure this in bang, bang, bang. Get your 3 fingers up for your 3 points, go through each point, move on. Everyone has a different style. You need to find your own and practice, practice, practice. Hopefully Tom's going to come back and do a little bit more Viva action. But if you are preparing for the Viva and you want to get on this show, be put through your paces, get some direct feedback, and obviously fame, then please email me, direct message me on Blue Sky, message me on Facebook, send a carrier pigeon, less likely to work, or, leave a comment on the show. And in the meantime, buckle up for the next episode of Gas, Gas, Gas. We're going to return to some core science stuff, the final element of the pharmacokinetics of propofol sort of series. We're going to talk TIVA, and we're going to have a dive into the various TIVA models, the differences, the pitfalls, and the things you should know. Cheerio, and see you next time! Thanks for listening to that episode, guys. If you found it useful or awful, please like and subscribe and rate the show. Definitely go check out those show notes on gasgasgas.uk. We all know that this is a bucket of content. I want you to take some time for yourself and don't overcook it. Don't freak out. Keep studying.